Okay, we are reviewing DNA structure and replication. So, as a review, DNA is your genetic material. It's stored in the nucleus of every eukaryotic cell. All cells of an organism have all of your organism's DNA, so that means your brain cells, for example, contain information for your liver cells. Your liver cells contain information about your heart cells. Okay, and you have 4 base pairs, A, T, G, and C. A always base pairs with T, G always base pairs with C. So the replication of DNA is said to be semi-conservative, which means that you start with 2 strands. You start with 2 strands that are hydrogen bonded together. They break apart, and each parental strand serves as a template for a daughter strand. Okay, so let's go into a little bit more detail with that. And I think you're saying to yourself, Mrs. Berger, holy smokerinos, there is a lot of stuff there. Yeah, there is. So I'm going to just break this picture down for you into individual steps. Okay, so the first thing that happens is your helicase, your unzipper. You get it right here. This helicase right here takes your double-stranded DNA and unzips it and opens it up to a fork, a replication fork. Okay. And to keep those base pairs from rejoining each other, because remember, let's pretend this is an A and this is a T. Let's pretend this is a G and this is a C. They're going to be attracted to each other, so they're going to want to bond back up. So these single-stranded bonding proteins, they're going to keep them apart, which is good. But it can't hold apart an entire DNA strand, so that's why we can only unzip a certain amount at a time. Okay, now DNA does have directionality to it. Okay, so let's say that this upper strand is running 5' to 3'. So that means that this strand is 3' to 5'. Okay, now this is important because we're going to end up with a situation where DNA polymerase, we'll get over there for a second. DNA polymerase can only synthesize in the 5' to 3' direction. Okay, so primase, the first thing that happens is primase bonds. And primase adds RNA primers to say, hey, this is where you're going to start DNA polymerase. Because DNA polymerase can only add nucleotides, it can't start a brand new strand. And they will be replaced with DNA later on. Okay. And I realize now that I mislabeled these. I'm going to make this upper strand just because of the things that I'd done before. Where you put in there, this upper strand actually runs 3' to 5', and this lower strand runs 5' to 3'. Okay, so now it's looking the same here. So this is 3' to 5' on our parental strand. That means our new strand, the one that's coming in to bond with that parental strand, this strand here is going to be my leading strand because it is able to run in the 5' to 3' direction, which is the same direction that DNA polymerase is able to work in. So that's going to be my leading strand. Now, this strand here, the lower strand, is 5' to 3'. Now, DNA polymerase can only work in the 5' to 3' direction. So what's going to happen is, let's say that this, we have 5' to 3' here. Okay. What happened before, just a few minutes ago, before this picture was taken, was that this was where the fork was, and this DNA polymerase moved its way down and produced this small fragment here, this Okazaki fragment. Okay. So let's keep going. So, and as this replication fork moves further away, you're going to see that this DNA polymerase now, whereas this one, the one on the top, can just keep on going, this DNA polymerase, it's heading in this direction, right? So in order for it to continue to make this strand down here, this DNA polymerase right here has to, I'm going to get my purple back, get myself a purple marker, has to hop up here and start again. Okay, so that's why that's called the lagging strand, because it has to start over a whole bunch of times. Okay, it produces what's called Okazaki fragments, these small pieces of DNA on our lagging strand. Where on our leading strand, we don't have those because our leading strand can just keep on going. Okay, but because polymerase can only work 5' to 3', we have to make, again here, another strand, another Okazaki fragment that are eventually going to need to be smoothed over by DNA ligase.